In this lecture, I am going to go over the Calvin cycle, which is the last of the steps in photosynthesis. Specifically, we will talk about the process by which carbon dioxide is fixed from inorganic form into organic form and eventually built into sugars. We will remind ourselves that the electrons and energy source for this are NADPH and ATP respectively, which both come from the light-dependent reactions in photosynthesis. We will talk about the first step of the Calvin cycle in detail, and this is where the enzyme Rubisco is used to combine the substrates RuBP and carbon dioxide together, and that's going to result in two molecules of something called PGA. And we will specify that the Calvin cycle fixes exactly one carbon molecule in each turn of the cycle, so it has to turn a lot to make glucose. Then we will talk about the fact that this entire process of the Calvin cycle is inhibited in its very first step by oxygen, and we'll talk about what happens if oxygen interferes and a process called photorespiration happens instead. Finally, we will talk about an alternative start to the process of the Calvin cycle, some steps that come in before, called C4 photosynthesis. And these steps help to reduce photorespiration. Let's take a look here at the general process of the Calvin cycle. It is shown in this diagram, and you can see that we can split this cycle into three steps. The first stage, really I should call it, is where carbon dioxide comes in. So this is the top of the diagram, and you can see that the molecule RuBP is being combined with carbon dioxide, resulting in PGA. So those are the carbon fixation step. Next, we have the step where the molecule that results, PGA, is built up into a higher energy compound, and we add some high energy electrons. And so this is called step 2, the reduction of PGA. And finally, you'll see that the last set of steps, or stage 3, is where some of our molecules that result from stage 2, namely G3P, are used to regenerate RuBP. And this is necessary so the RuBP will be available to do another round of the cycle. You can also see down here at the bottom that some of our G3P is going to exit and be used to form glucose. So now let's go over that in much more detail. Let's talk in detail first about stage 1, the fixation stage. Here we have carbon dioxide being added to RuBP, which is a 5-carbon compound, and this is going to happen by the enzyme Rubisco. And so take a second just so you can count and confirm that RuBP in fact has 5 carbons, and you can also see it has a couple of phosphate groups. Carbon dioxide, of course, has 1 carbon. When those come together, they are going to form a 6-carbon compound. However, that 6-carbon compound immediately splits in half and forms 2 molecules of 3-carbon compounds. So 2 times 3 equals 6. And each of these molecules is the same. They are both called PGA. We'll go over this in even a little bit more detail in just a second. But what we've accomplished here is adding an inorganic carbon in carbon dioxide into organic form once it is incorporated as part of one of our PGA molecules. So now I will go over this in even more detail with specific names, and I often use this as a bonus question on the second exam, where I will ask students to tell me what are the full names of the reactants, products, and enzyme in the first step of the Calvin cycle. So this RuBP, if we take that abbreviation apart, what we're really looking at is, ribulose 1,5 bisphosphate And why is it called that? Well, we won't worry about the name ribulose, but you can see there's a phosphate group attached on the 1 and 5 carbon. And so that is where the 1,5 bi or bisphosphate name comes from. Of course, you know carbon dioxide. This is going to result in 2 PGA molecules. PGA is phosphoglycerate, and it's 3-phosphoglycerate because the phosphate group here is attached to the third carbon. Finally, we should specify what is the full name of this enzyme. It is ribulose 1,5-bisphosphate, because that is one of our substrates, carboxylase oxygenase. So the carboxylase in this name is referring to the fact that it is fixing carbon in the form of carbon dioxide. We'll talk about what the oxygenase means in a few minutes. So now let's go on and talk about what happens after we've incorporated carbon dioxide and made PGA. That's sort of the important part, because the whole goal of the Calvin cycle is to build organic molecules, and adding the carbon dioxide is doing just that. But we need to take this PGA and make it into a glucose molecule. So we are going to do that mostly in the reduction stage. So in the reduction stage, over here, we have our PGA, but we are going to do some rearrangements. They are energetically expensive, and we will pay for them using ATP. So these are energy coupling reactions where ATP loses energy, a phosphate group is released, and it becomes ADP, but meanwhile, the molecule PGA gains energy as it is rearranged. Additionally, we need to add some electrons associated with high potential energy. We stored those in the late dependent reactions on NADPH. So now this is where we will use that NADPH. We will oxidize it, remove those electrons, and transfer them to this 3-carbon compound that we are building. Once we have done these rearrangements and added the high potential energy electrons, we will end up with a molecule called G3P. G3P is a name you should recognize. And you should recognize it specifically from being a molecule that occurred about midway through glycolysis. Remember, this is glyceraldehyde 3-phosphate, and remember during glycolysis, glucose was broken down and formed 2 of these. And so what we're doing here is basically setting ourselves up for the reverse process, where once we've formed G3P, we can take 2 of them, put them together, and form a single glucose. So you can see there are 3 carbons in each G3P. You know that there are 6 carbons in glucose. So that's why 2 of them would give us a single glucose. And so by the end of the stage 1 and stage 2 of the Calvin cycle, we have G3P and we could make glucose. And that's great because our goal in the Calvin cycle is really making glucose. The problem is that we need more RUBP, that first molecule that we used as a substrate in stage 1. If we run out of RUBP, then the Calvin cycle has to stop. And so what this means for the plant is that in practice it cannot use all of its G3P to make glucose. Instead, it is going to use most of it simply to regenerate RUBP so the process can go forward. I will point out here that the book uses a different abbreviation. It calls this 3-carbon molecule GA3P. It's the same thing, but G3P is a simpler and I think more common abbreviation. So we'll just use G3P. Second, I want to point out a couple of things about this regeneration process. You can see that G3P has 3 carbons, and we've already said RUBP has 5 carbons. That makes it really complicated to regenerate RUBP from G3P. And the math works out like this. If we can take 5 G3P molecules and combine them together and rearrange, 5 times 3 carbons is going to give us 15 carbons to work with. You can then see that with those 15 carbons, once we've rearranged them, we could make 3 molecules of RuBp because that has 5 each. So basically our math is 5 times 3 equals 15, and that is going to give us 15 divided by 5 is again 3. The next thing I want you to notice is that in this regeneration process, it requires additional energy. So we are going to expend ATP here, again, creating some ADP. So if you recall back in the light-dependent reactions, we talked about cyclic and non-cyclic photophosphorylation. And we said that the cell needs more ATP than it does NADPH. Here we can see part of the reason. We use NADPH only at one stage of the Calvin cycle, but we need ATP in 2 different stages. I am not very concerned here about the number of ATP or NADPH that are required. I mostly want you just to recognize what is required in which steps. We've said that most of the G3P in the Calvin cycle is used to remake RuBp, but not all of it actually is, because of course we do need to make some glucose. So here's how you can think about how much gets diverted from the Calvin cycle. As this is going around, each time it happens, we are fixing 1 carbon because we are adding 1 molecule of carbon dioxide. Ignore the 3 up here and just think about 1. So if we add 1 carbon dioxide each time, then that means every 3 times this cycle happens, we've added 3 carbons. Because G3P is a 3-carbon molecule, this means that every 3 times through, we have accumulated enough fixed carbon that we can remove a single G3P and use it to make sugar. Since we said that glucose has 6 carbons, that means it's really 6 times through this cycle that is necessary for enough carbon dioxide to be incorporated that we will have adequate carbon to make an entire glucose. So 3 times through the cycle for a G3P, 6 times through the cycle is enough to give us 1 glucose molecule. We specify the 3 cycles for G3P, but you can add the 6 equals 1 glucose as well. And this is just that same slide, but I've blown up the image so you can see the details in case it was hard to read. What we need to talk about next is a little bit more about the enzyme Rubisco, and we need to talk about the second part of its name. So remember we said Rubisco is actually an abbreviation for ribulose 1,5-bisphosphate carboxylase oxygenase. Carboxylase because it incorporates carbon dioxide, oxygenase because it can instead, if the plant is unlucky, incorporate oxygen. So let's think about why this is unlucky. First, if it incorporates carbon dioxide, that is just the normal Calvin cycle. That's what the plant wants the Rubisco there to do. But oxygen also fits into the active site of Rubisco. In other words, oxygen is a competitive inhibitor of the carboxylase function of this enzyme. It goes into the active site, it prevents carbon dioxide from doing so, and the oxygen is then instead used as a substrate. This process, when oxygen comes in instead, is called photorespiration. Unlike the Calvin cycle, photorespiration does not result in sugar production. So let's think about what happens. If we look back at the Calvin cycle, imagine that an O2 molecule came in instead of a CO2 molecule. Well, then we have a 5-carbon compound, but we're not adding any carbon, so all we have here is 5 carbons. But that molecule that results will need to be split in half. It can't split into 2 3-PGAs because we don't have enough carbon. So the plant now needs to do some alternative metabolism to try to regenerate RuBp, even though it is not starting with 2 3-PGA molecules. Plants, in fact, do have a way to regenerate RUBP molecules if accidentally oxygen is incorporated instead of carbon dioxide. It's a complicated series of reactions. It involves 3 different organelles, including the chloroplast, and it requires rather than bringing in carbon dioxide, it actually causes the release of carbon dioxide. So this is a problem. Remember, the whole point of photosynthesis is bringing in carbon dioxide. Instead, we are doing just the opposite and releasing it. Even worse, this process requires ATP. And so the plant is both losing carbon and it is expending energy. As a result, photorespiration is a big problem for a plant. Let's talk for a second about why this is called photorespiration. Think about respiration that we do or that plants do with their mitochondria. In that process, oxygen is incorporated and carbon dioxide is released. In this process we just talked about, with photorespiration, oxygen is also incorporated and carbon dioxide is also released. And so by analogy, it is similar to respiration that we've already studied. However, respiration, as we've studied it, results in generation of ATP, so when the cell is able to store energy. In photorespiration, the reverse happens, and ATP is expended or broken down instead of being formed. So while this has similarities to cellular respiration, it is not resulting in the useful product. Overall then, photorespiration is a process plants want to avoid. Photorespiration is especially a problem in hot, dry environments. Let's talk about why. In a moist environment, the plant has plenty of water, and so it can keep its stomata open and allow gas exchange. So if there's lots of water around, then if stomata are open, carbon dioxide can diffuse into the leaf, oxygen can diffuse out, and of course some water will be lost, but the plant can replace that from water it collects from the soil. However, if the soil is dry and it's a hot sunny day, then the plant needs to prevent water loss. It can do that by closing its stomata. So the guard cells close, this opening closes. That will prevent additional water loss. However, it will also prevent carbon dioxide from diffusing into the leaf, and as photosynthesis happens, oxygen will build up. As that oxygen builds up, if the stoma is closed, it will be prevented from diffusing out of the leaf. That means on hot, dry days, the carbon dioxide concentration inside the leaf diminishes or goes down, while the oxygen concentration accumulates or increases. As a result, on a hot, dry day, initially maybe the Calvin cycle will happen, but as the day progresses, photorespiration will overtake the Calvin cycle and the leaf will start losing more carbon than it gains. Plants in hot, dry environments have an alternative process to deal with this. There are 2 closely related processes, one called CAM photosynthesis and one called C4 photosynthesis. We will focus on the C4 photosynthesis. We said this happens in hot, dry environments, so this is where we find most plants that have this process. Not all plants can do it, only a subset of plant species are capable. And the basic idea here is that the plant is going to add some additional steps prior to the Calvin cycle. And these additional steps are going to separate the process of acquiring carbon dioxide from the process of fixing it into organic form in the Calvin cycle. We're going to see that the process is spatially separated between 2 cell types. And the important cell types here are the bundle sheath cells. In dark green, they're surrounding vascular bundles, or basically the veins of the leaf. And the cells that surround those are called the mesophyll cells. We'll see they each have a different role, One will be for capturing carbon dioxide, the other will be for doing the Calvin cycle. So let's go in and look at a more detailed version of this. In the first step of this process, it is going to occur in the mesophyll cells. So the mesophyll cells are these outer ring cells. And in those cells, the plant will capture carbon dioxide. The carbon dioxide will diffuse in, and it will be incorporated into an organic compound called oxaloacetate. So you can see that here. The substrate for this reaction is a 3-carbon compound called PEP. So you can see 3 carbons represented, named PEP, and it is going to combine with CO2, and that is going to generate the 4-carbon compound oxaloacetate. The enzyme that does this reaction, remember that it, like all other enzymes, is named after what it does, and so its name is simply PEP carboxylase. So what have we accomplished so far? We have captured carbon dioxide and made a 4-carbon compound. The nice thing about this step is that the enzyme PEP carboxylase has a high affinity for carbon dioxide, but it has no affinity for oxygen. So while Rubisco is inhibited by oxygen and can do photorespiration, basically PEP carboxylase just ignores the oxygen no matter how much has accumulated in this mesophyll cell. So now let's go on. We've got our 4-carbon oxaloacetate, and let's see what happens. It will be transferred through a plasmodesmata, or I should really refer to it as a plasmodesma singular, but either way, it is transferred through an opening into the adjacent bundle sheath cell. Here, our 4-carbon compound is going to break apart again. One of those carbons is going to be released as carbon dioxide. Basically then what the plant has accomplished is transporting carbon dioxide from the mesophyll cell to the bundle sheath cell. A lot of carbon dioxide is released by a lot of oxaloacetates in the bundle sheath cell, and so the bundle sheath cell ends up with a high concentration of carbon dioxide. Meanwhile, once this carbon dioxide is released, the remaining 3-carbon compound is cycled back into the mesophyll cell. It needs to be modified a bit to be modified back into PEP, and then it can again capture a carbon dioxide, form oxaloacetate, and be cycled back. And so this compound is basically going to continuously cycle back and forth from the mesophyll to the bundle sheath cells. And we are going to be able to maintain a high carbon dioxide concentration in the bundle sheath cell. The bundle sheath cell then is going to simply do the normal Calvin cycle. So once the carbon has been released, we have a high carbon dioxide environment. That means that the enzyme Rubisco is going to be very likely to encounter carbon dioxide instead of oxygen inside this cell. And as a result, the Calvin cycle is not going to be strongly inhibited by photorespiration. The normal Calvin cycle will be able to go forward at a high rate. So what this then allows is, even in a hot, dry environment, if the plant has this setup and it has PEP carboxylase, then it will be able to continue doing photosynthesis even as the plant has to close its stomata. So that then concludes what we need to say about the Calvin cycle and C4 photosynthesis. Let's just remind ourselves of some important points. First, the Calvin cycle is the process at the end of photosynthesis where we actually build G3P, which can be used to make sugar. Second, I tell my students they need to know the details of that first reaction in the Calvin cycle. The rest of it, they just need to have a conceptual understanding at the level I talked about here. Third, remember that the raw materials, specifically the NADPH and the ATP, are both coming from light-dependent reactions. So while the Calvin cycle itself does not use sunlight, sunlight is nevertheless required because sunlight is necessary to make ATP and NADPH. Number 4, for each turn of the Calvin cycle, one carbon is fixed into organic form. That means 6 turns are necessary to make a single glucose. Then, with regard to photorespiration, you need to know that oxygen is a competitive inhibitor for the enzyme Rubisco, and when oxygen is incorporated by this enzyme, the process of photorespiration rather than the Calvin cycle follows. And this is bad for several reasons. It requires ATP, and it results in the release of carbon dioxide instead of the capture of it. And finally, some plants that, especially those that occur commonly in hot, dry environments, can do an alternative process called C4 photosynthesis, which separates the initial capture of carbon dioxide from the Calvin cycle. And as a result, it is able to reduce photorespiration by separating these two processes.